excited to talk to you today about our use of different DNA sequencing technologies to assemble and characterize metagenome samples. And even though I'm giving you this presentation today, this was a major collaborative effort. I'd like to, even though I will acknowledge more members of our consortium later, I'd like to give specific thanks and gratitude towards our collaborators at UC Davis, Misha Kolmogorov and Pavel Pesner, as well as our collaborators at PacBio, specifically Elizabeth Zhang, and also Tim Smith at USDA MARC. I've sectioned my talk into three distinct topics. The first, I wanted to give some background information on what a metagenome is and what its use is in the research community. Then I'd like to talk a little bit about the complexities of assembling the metagenome and the limitations imposed by DNA sequencing technologies. Finally, I'd like to talk about a big breakthrough that I feel will change the field of metagenomic sequencing and assembly, and to conclude my talk at the end. But the most important thing is, for this talk's purpose, what is a metagenome? And I will be using the following definition, which is the totality of microbial genomes derived from, in this case, a tissue. And I'd like to state more specifically that when I'm talking about a metagenome and a metagenome assembly, I'm talking about the DNA sequence within the sample, and even more specifically, just for clarity and illustrative purposes, just the DNA within single-celled microscopic organisms. This is not always the case. There are some multicellular organisms within metagenomes, but their means of classification and annotation are still novel. The metagenome has major impacts on a diverse array of different subjects in the field of biology. Much of this is derived from experiments we've done on gnotobiotic animals. These are animals that are reared in abiotic environments and are deprived of their innate commensal microbial communities. We found that such individuals, particularly mice, have smaller large intestines and can often be malnourished if given standard diets due to lack of vitamins and coenzymes. In addition, metagenomes have been found to play a major role in vaccine efficacy in mouse models. It turns out that the certain cell surface receptors in the gut need to be stimulated by the commensal microbial community to elicit immune responses against certain pathogens when given a vaccine. And finally, I would like to point out that even though a lot of our data comes from these sorts of model organisms, metagenome complexity and influences exist across the entire spectrum of environmental and living species. There are many symbionts and competitors that will actually cause changes in growth of many different species based on the presence and or absence. But even though the metagenome can be important in these fields of study, it's a generally temporal and diverse set of organisms. And this causes a great deal of complexity and confusion in terms of how it can be assessed. For example, there can be different sampling biases when you're trying to assess the metagenome community based on the presence of different organisms that are recalcitrant to DNA extraction or sequencing. And in addition to that, a metagenome is often a snapshot in time when it is sampled. And much of this has to do with the fact that certain organisms can be present in one stage or condition and absent in another, and this can vary in the order of seconds or minutes or hours. In addition to that, these are living organisms that reproduce and as such sustain mutations as they pass down their genetic information. And so this sort of variation needs to be carefully distinguished and characterized in any metagenomic sampling. There are some different ways in which we can characterize the effects of metagenomes on systems and environments. Probably the simplest and most straightforward would be to measure both inputs and outputs from a metabolic standpoint. However, this sort of treats the metagenome as a black box, and distinguishing characteristics of the metagenome are lost in this translation. One method that is the gold standard is to use amplicon sequence analysis, where you target conserved genes that should be present in all lineages, amplify them, and identify variants. This one common gene that's targeted for this sort of analysis is the 16S subunit ribosomal sequence. And the reason for this is because it's prevalent in almost all prokaryotes and has very well-conserved regions flanked by variable regions that sustain mutation. And the final and most in-depth is to use whole genome sequencing and assembly. In contrast to the aforementioned amplicon sequence experiment, this is more expensive and requires a lot more computational analysis. However, it gives a better picture of all of the genomic DNA in the system as opposed to the use of a single copy gene and extrapolation based on variant analysis. Even though 16S amplicon sequencing is the gold standard and often is used statistically to claim high degrees of completeness, there are some environments in which we have limited knowledge of the system in order to classify variants and distinguish lineages. And one such example, taken from a whole genome shotgun sequence analysis in the rumen, published by Stewart et al. In 2017, they attempted to take genome sequence reads and classify them based on genomic databases. When they used all previous assembled microbial genomes, including all of RefSeq, in order to classify the origin of the reads from their study, at max they had a 30% classification rate, which meant that 70% of the reads had never been seen before in previously used databases. It was only when they took those reads and generated the assemblies themselves for a sample-specific assembly that they had higher classification rates and were able to assign those reads to different lineages. However, DNA sequencing analysis is commonly contrasted against the activity of solving a jigsaw puzzle. And as you can imagine, the old trick that's commonly given as advice for starting jigsaw puzzles, where you actually take a look at the box art, is not applicable here. We don't know what the composition of the community is. However, we are able to rely on computational algorithms called genome assembly algorithms to use different segments of DNA in order to reduce the complexity and try to find a better resolution and more details of the picture of the metagenome. Typically, these data sets consist of hundreds of millions of pieces, so it would be impossible for you to manually solve a metagenome puzzle within the span of your life. And to give more clarity and terminology, the algorithms that we use are termed assembly algorithms, and you can think of the most simple sort of algorithm and most intuitive as being the comparison and overlap of DNA segments. This type of DNA assembly algorithm is called consensus overlap, and the resulting sequence is called a contig, or a contiguous stretch of DNA. A slightly less intuitive method is the use of assembly graphs, where you take the DNA sequence of each read and decompose them into a graph that can be fed forward and improved by the addition of other reads. This is the algorithm that's used by the Metafly assembler that we use in our proof of principle later. And finally, you cannot typically generate a single microbial genome assembly from a contig. So we use long-range information in order to try to take a bunch of contigs and place them together in what we assume is the genome sequence of an organism from the community. And special information is required for this, including the use of linkage information that I'll talk about later. However, as I mentioned, DNA sequence cannot typically be used to form an entire contig, and much of this is because of errors or issues with the complexity of the genome. For example, the read length needs to be larger than repetitive sequence, and because the presence of orthologous or highly similar genes in different organisms, you need to have highly accurate reads in order to identify and distinguish them. We found this in our recent study in the rumen using a comparison of long and short read DNA sequencing technologies, And even taken from the same sample at the same time, a completely different picture was found in terms of both the reads themselves on the left of the diagram and the assembled contigs on the right. As it turns out, we identified that the long reads were able to better extend the contigs and create longer contigs in general. However, because of the lower error rates of our short reads, we were able to identify and assemble organisms that were at lower depths of abundance in the metagenome. To give you a brief overview of DNA sequencing technologies, I picked 5 of the most common technologies and compared them based on their read lengths on the x-axis. The first two, Sanger and 454 sequencing, are no longer used in the analysis of DNA sequence, mostly because they don't provide additional benefits compared to the other technologies. Illumina short reads still provide substantial benefits in terms of the economy of cost, and also have very low error rates, making them suitable for variant calling in samples. And the two latest technologies from Pacific Biosciences, or PacBio, and Oxford Nanotech introduce longer read lengths than any of the prior technologies. However, this comes at greater cost and have higher error rates compared to the Illumina technology, typically. But I'd like to talk a little bit more about the PacBio library prep and how this can be used to correct for that Achilles heel in the technology. So starting with a genomic read segment at the top, if you go through the normal PacBio library prep, you anneal 2 hairpin adapters on the side of the read, effectively circularizing it in a single strand. If you were to take one single sub-read or one single segment of the read and sequence it, you'd still have about a 14 to 17% error rate, mostly through the addition of indels. However, the read is circularized. So if you're able to read through the same molecule multiple times, given that it's from the same molecule, you can use all of those reads to compare against each other and create a consensus. And the brilliance of this is that by taking the same molecule, you can actually reduce the error rate lower than 1%, giving it parity with the shorter read technologies. However, not abandoning the longer length of the read. In addition to this, we also wanted to generate some longer range information, and we typically use Hi-C data in order to generate this for metagenomes. Hi-C is the use of cross-linked DNA within individual cells, and in this figure you can see the cross-linking indicated by the black bars. After you cross-link and shear the genome, you can create sequencing libraries that preserve this long-range interaction and generate a data set for you to use downstream. Using the statistical association of those junctions, you can actually take contigs and associate them with the DNA that was predicted to be in the same cell at the time of cross-linking, thereby binning contigs into genomes. And the ultimate goal of both binning and assembly is to create resources that allow you to classify metagenomes. And we assess these through 3 different criteria. The first is through the continuity of the assembly, which is the length of the contigs themselves. But we can also assess completeness and potential misassemblies by counting the number of single copy genes that are expected to be in each lineage. Based on the proportion of those genes, you can estimate how complete a bacterial genome assembly is. Likewise, if you find more than one copy of a single copy gene, that indicates the potential for a misassembly or the inclusion of data that should not be there in that genome. But there's another caveat, and that is microbial lineages within the metagenome can be quite diverse, and we're not often able to resolve them down to every specific lineage. So if we take this dendrogram and we divide it up based on taxonomy, we typically divide it based on the percentage of sequence that's shared amongst lineages. And you can see that a genus, a species, and a strain are in decreasing order in terms of their resolution. In all practical purposes, though, when we finally create our final set of bins, which are typically termed microbial assembled genomes, they are composites of the lineage and are often not resolved down to the strain level. So in many cases, a MAG may be a composite of a species or even a genus in the end result. So I'd like to shift focus now and talk about our proof of principle study. And in this study, we wanted to do a full-length characterization of a single sample taken post-mortem from a sheep. And we sequenced it with several different DNA sequencing technologies to incredible depths, and then assembled it using the Metafly assembler. To give you a sense for the amount of data that we collected, the primary DNA sequencing technology was the generation of PacBio hi-fi reads, which were in total about 255 billion bases of DNA sequence, or gigabases. In addition to this, we generated Illumina short reads for both read depth and polishing purposes, and we also generated Hi-C short reads for binning. And we wanted to ask, what are the explicit benefits of these low error rate long reads in metagenome assembly? Can they overcome the challenges that we've identified? And we wanted to do an apples to apples comparison because we realized we can't really compare the unique benefits of low error rate if we take different samples and sequence them using different technologies. So I think we found a clever way around this to provide that apples to apples comparison. So in this diagram, I show a schematic of the PacBio read in multiple subreads. Just as a reminder, each of these subreads is used to correct the read for the final hi-fi data set. So this entire read is turned into one read in our hi-fi data set. But I thought, what if we could actually get around the sampling bias, where if we sample the same, if we sequence the same sample multiple times, we may get different results. What if we took individual subreads from that same sequencing run and used them as if they were uncorrected PacBio reads? This now allows us multiple samples, multiple low uncorrected reads that we can use in our comparison. So what we did was we took each of those data sets and assembled them using Metafly in order to compare against an equivalent amount of uncorrected long reads versus an equivalent amount of hi-fi corrected reads. But first, I'd like to show you the hi-fi assembly, which was incredibly high quality, as you'd expect. We identified 49 closed circle contigs just from the initial assembly, and this was without any additional work or analysis to generate them. You can see the larger hairball in the middle of the graph, and this turned out to be an amalgamation of both eukaryotic genomes, which we didn't characterize in depth, and also prokaryotic sequence. But you may also notice that there are smaller circles in this graph, and these represent what I believe were found to be satellite DNA, such as plasmids. Returning to our comparison of the CLR assemblies, I classified each of the contigs based on its predicted taxonomic affiliation based on kingdom. And you can see here the plot of the total length of the assembly in each of those categories, from viruses, eukaryotes, archaea, and bacteria, and also those that were unclassified in the no-hit category. In all cases except for the unclassified category, the hi-fi assembly had 10 to 35% more assembled sequence of that superkingdom. But it's not just longer amounts of DNA sequence for each kingdom. The hi-fi assembly was actually more complete as well. This is a graph of the completeness of these contigs based on the count of single copy genes. Anything that's above 90% is considered to be incredibly high quality draft genomes. And you can see from the hi-fi data set, it's over twofold higher in greater than 90% completion than the other assemblies, and actually has the highest number of contigs at this category than anything else that's been published in the literature to date. So in order to improve this incredible foundation, we used Hi-C data to try to create a final set of bins or mags from these contigs. When we did so, we found a very interesting phenomenon, and that was the fact that when we compared the average X coverage of short reads to each of our bins against the cumulative length of our assembly, we noticed a great discrepancy. So you'd expect that as the assembly approaches the right-hand side of this graph, it would actually increase in length. And the lower ends of the graph would indicate that less abundant lineages are being assembled. And this was the case for the hi-fi assembly. We actually found that 16% of the length of the hi-fi assembly was in bins that had less than 5x coverage of short reads. But does this mean that we're just assembling a bunch of low abundant species and they're not really resolved very well and not very complete? When we take a look at our bins based on single copy gene completion at the greater than 90% category, we actually find a greater percentage of hi-fi reads at less than 5x coverage than the CLR data sets, as well as several hi-fi bins that have less than 1x coverage, which shows incredible quality at distinguishing these lineages. So the question is, are the CLR assemblies just not assembling the low abundant lineages? It turns out we don't think that that's the case. And in this bipartite graph, I want to show a comparison between the data set from CLR1 to our hi-fi bins. And in this graph, we're actually showing association between bins from the CLR side to hi-fi that indicate that they may be orthologous genome assemblies that have been split based on matched distances. The edge color indicates the number of associations, where purple indicates 2 hi-fi bins for every 1 CLR bin, whereas yellow indicates 3 for every 1 CLR bin. We found across all 3 CLR data sets, there was an average of 12 CLR bins that tended to be these collapsed assemblies of at least on average 26 hi-fi bins. But in particular, I'd like to point out one example that I think is very instrumental in understanding why this is occurring, and this is for our yellow edge for CLR bin 451. When we compare the assembly graph generated by Metafly, it's quite clear why this lineage is poorly resolved in CLR. This is the hi-fi assembly graph, and each of the hi-fi bins that were composites of this are labeled in different colors, with blue being hi-fi bin 451, orange 452, and green being 471. You can see that even though they have similar regions that result in the connections between all 3 bins, you can sort of pull them apart yourself and create each separate bin yourself. When we take a look at the same composite CLR bin, you can see why this has all been condensed. Using CLR reads, we were not able to distinguish between those 3 distinct lineages. And this is primarily due to the error rate of the reads. When we align hi-fi reads to our hi-fi bins, we can also see a consistency in read depth. The read depth is the blue bar, and the depth of coverage for each bin was 10x, 20x, and 30x, respectively, for the 3 hi-fi bins. How does this look in CLR1 bin? It's highly variable, and the average depth is around 45x, which suggests that this is indeed a composite of those 3 bins. Now, keep in mind, all 4 of these bins were considered to be high completion and low redundancy. So it just goes to show that this sort of resolution is integral in actually distinguishing between these lineages, and not all high-quality bins are created equal. And this gave us great confidence in using hi-fi reads for other sorts of analysis. And one particular area that we thought that hi-fi reads could provide substantial benefit is in phasing variants that we identify in the sequence data. Phasing is the use of statistics or physical data in order to link separate variants. In our case, we're using the hi-fi reads themselves. And to illustrate this point, this diagram shows 2 different read sets in both red and blue that phase different single nucleotide polymorphisms at the top into distinct haplotypes. A haplotype is a linked set of genetic variants that will be passed on to descendant lineages. And we can actually distinguish these using the hi-fi reads, whereas it was far more difficult to use short reads to do the same. So we are the first group that attempted to apply this to metagenomes, and we've created a tool called MagPhase, which exclusively uses hi-fi reads to identify and distinguish haplotypes in metagenomes. This is a table of the results from MagPhase, and what we were able to find is that MagPhase was capable of linking and phasing variants up to 500 kilobases in length, which is a substantial proportion of a typical bacterial genome size of 4 megabases. But the most important values I'd like you to look at are the percentage of polymorphic mags which had at least one identifiable haplotype in our hi-fi data set. You can see that the majority of our hi-fi data set, our hi-fi mags, do not have polymorphic haplotypes that could be discovered, whereas in the CLR data sets, given all the data that I've shown so far, is consistent with them being collapsed and condensed representations of the metagenome, and had higher percentage polymorphic mags. I'd also like to point out that in some regions of certain mags, there were high degrees of polymorphic alleles. And we actually counted up to 25 alternate haplotype alleles in some regions of the hi-fi assembly. This was double in the same or equivalent regions in the CLR data sets. Again, these were composites of highly polymorphic regions. A picture's worth 1,000 words, so our collaborators at PacBio helped us in generating these IGV plots. And you can see at the top in the red circle, the thick red square, that we actually have depths of coverage of different variant sites indicated by alternate colors. And these are clearly distinguished within the hi-fi data. You can see the alternate allele is patterned here, and the reference is patterned underneath. And these are appreciable depths of coverage that are pretty consistent across the identified variant sites. When we compare the same region in an orthologous CLR lineage, you can see the picture gets occluded quite quickly. There are at least 3 different alternative haplotypes that were identified by MagPhase, and much of the reason for this identification was a high degree of heterogeneity in regions upstream and downstream of the target sites. It was very difficult to distinguish because of the high degree of variance and the low resolution of the sequence in this region to distinguish between them. So, in conclusion, the complexities of metagenome assembly are quite daunting, but the utility of knowing the metagenome has great impacts on many different types of research projects. We found that hi-fi reads provide substantial benefits to metagenome assembly and additional utility. And we've also identified a means by which you can use hi-fi reads to phase metagenome lineages through the discovery of haplotypes. And we are planning to release the software through publication fairly soon. And I'd like to acknowledge many of our collaborators. This was a large collaborative effort, particularly with my colleagues at the USDA, Tim Smith in particular, our colleagues at UC Davis, in particular, Misha Komogorov and Pavel. PacBio is incredibly instrumental in the development of MagPhase, and all of our other consortium collaborators. Since I'm with the USDA, I have to provide a disclaimer. The mentioning of trade names or products is for informational purposes only, and the USDA cannot endorse specific products. Thank you very much for your time, and I'd be happy to take your questions.